Okay, I want to make a few announcements for you. The first announcement is that Bio 1B is an impacted class. It's a class which is oversubscribed as far as the number of people who want to get in. And as a result, it has a very early drop date. So the drop date for this class, if any of you want to drop it or at home want to drop it, is this Friday. If you wait until after this Friday to drop the class, you have to make an appointment with the dean, and I think you also have to pay something to drop the class. So again, for those of you who are, for whatever reason, not going to continue in the class, you'll need to act by Friday, or else it'll make your life a little more complicated otherwise. The second point I want to make is that several of you have asked me about the role of a textbook in the class, and specifically in the plant section. I consider that the exam questions will come exclusively from lecture. The textbook, which in the plant section I don't think is very good, is mostly represents a chance for you to read up on an area that I may have skipped over or gone too quickly over. But if I haven't mentioned something in lecture and it's discussed at greater length in the textbook or discussed a lot in the textbook, you needn't worry specifically about that being on the exam. I have assigned more pages than I actually will cover in the lecture, in part to give you some background, but also in part to let you know what pages correspond more or less to the topics of the lecture. Are there any questions about that? Yes?
Is the information on the handouts for the labs, is that also going to be covered?
Is the information on the handouts for the labs also going to be covered on the exam? It won't be specifically covered on the midterm, although your GSI will probably use that material, and it's possible that it's the same thing that I've covered in lecture. So the answer is in that way, yes. All right. Last time I ended by talking about the gymnosperms, which we said were the plants that produce cones. And we said they produce two kinds of cones. These woody cones that many of you are familiar with, which represent the cone-bearing plants, the firs and the redwoods and so forth. And then a more transitory type of cone, a much smaller cone, which was produced either on the same plant as produces the big cones or sometimes on two separate plants. What I'd like to do now is to return to these two cones to sort of re-emphasize some of the important points in the life cycle, and then finally to end up the portion on the gymnosperms, contrasting them with the group of plants which immediately preceded them, namely the seedless vascular plants. So I'd like to begin first by talking about the male cone. Now, we said these cones are made up of sporophylls. And we defined a sporophyll as a spore-bearing leaf. So what I'm going to do, and this is to help you orient, I'm going to make a longitudinal section. I'm going to cut down through the cone parallel to its long axis, and then draw that longitudinal section for you up on the board so that you can actually see where the sporophylls and the spores are located. So here is a longitudinal section, which we'll abbreviate as LS. Here is the main axis. Now this is the male cone that I'm going to draw. And the male cone has sporophylls. Sporophylls, all arranged along the axis. Now, it's a sporophyll because it has something to do with the production of spores. These drawings, by the way, are also in your outline. You can draw them again, or there's a little better drawing in your outline that you can print out. Let's look at one of these sporophylls. Attached to each sporophyll is a sporangium. And we said that sporangium or sporangia make spores. So inside, we have spores. This is the male cone, the tiny one that's not very long-lived. These spores, we said, are equivalent to pollen grains. And in the gymnosperms, we said that they are dispersed by wind. Now let's look at the female cone, one of these large cones, the one that produces the female gamete, which is the egg. And again, this should be a review from last time. Here is the axis of the cone. This is the axis. And here are the sporophylls. So this is the female cone, a longitudinal section of the female cone. Here are the sporophylls. And associated with each sporophyll is a sporangium. And now what I'd like to do is to magnify or enlarge a sporangium to talk to you a little bit more about it. First of all, let's take this sporangium here and let's just enlarge it. So we have an enlarged sporangium. Okay, the sporangium is surrounded by the sporangium wall. And we said this was a megasporangium, so it's a megasporangium wall. Megasporangium wall. And what is made inside of a sporangium? Spores. So in this case, we're just going to put one spore. And we call this a megaspore because it's made inside the megasporangium. And it is now N because meiosis has occurred. Here's the sporangium again. And now, what does a spore become? What do spores become when they germinate? They're haploid. What do they grow into? Gametophyte. What does the gametophyte look like in the gymnosperms? Well, if we look at the gametophyte, so this would be the female gametophyte. The female gametophyte. Inside, we would find an archegonium. And what is the gamete that is produced in the female? The egg. Right. And here we would have the egg. And this is all taking place on the scale. All taking place on the scale. Now, eventually, because these are the seed plants, we're going to form, although it hasn't formed at this point, this is the incipient seed coat. The incipient seed coat. Hasn't hardened yet, but it's beginning to form. This is this new structure that was added. When we combine the incipient seed coat, the archegonium with the egg, and the female gametophyte together, this is essentially the organism waiting to be fertilized. We call this structure, once it's preformed but not fertilized, we call it an ovule. An ovule. So when you have the egg formed and you are ready for fertilization, everything is at this point making an ovule. And now let's follow fertilization. The pollen is dispersed by the wind. Now, there are a couple of points which you should realize. We mentioned last time that the pollen grain is a microspore, and it is also equivalent to the pollen grain. When the pollen arrives by the wind, if it is in a right location so that it can fertilize, the pollen grain may undergo one or two more rounds of division so that the microspore has now become the mature microgametophyte. And it consists of only, probably less than 10 cells. So in this particular example, the gametophyte, that is the male gametophyte, is reduced to a few cells. Now, let's assume fertilization has occurred, and I want to show you the main components of the now developing new sporophyte or the seed. So at this point, we've had fertilization that has occurred. We now have the seed coat, which has formed on the outside. We may have remnants of the megasporangium wall. Probably hard to see. And if we look inside of this structure, filling most of the structure is now megagametophyte tissue. Megagametophyte tissue, except for one portion. And that portion in the very center, the product of fertilization, is the new embryo. The new embryo has formed in the center of this. So what I want to do for you now is to make sure you understand where the genetic material comes from to make several of these structures and what the genetic level is. First of all, what's the genetic level of the embryo? It's 2N, sorry. And it comes from who provides the genetic material for the embryo? Half from the mom, half from the dad. Okay, how about this megagametophyte tissue? What is its genetic level? N. And where does it come from? The mom only. How about the seed coat on the outside? Where does it come from? Comes from the mom, and does it come after meiosis has occurred? So it's 2N. It's not N. So we have the seed coat now, which comes from the mom, and it is 2N. So it's important when you look at a seed that you realize, especially the seed of a gymnosperm, that you realize the amount of the level of the DNA in the various portions of the seed and who's contributed to what. We're going to spend some time in the lab looking at this megagametophyte tissue because this megagametophyte tissue serves as a food reserve for the developing seed. And for those of you who eat pine nuts, and we'll give you examples of these in the lab, the nutritional material that you have in a pine nut is mostly megagametophyte tissue. That's what you're eating. And it's oily. And in the lab, you'll have a chance to open up one of these seeds, and in the inside of the seed, you'll actually be able to see the embryo, surrounded by its megagametophyte tissue. So now let's make a summary of the gymnosperms and how they differ from the ferns. This is an important summary. So the differences between the ferns, which were the immediate earlier group, and the gymnosperms. Remember, they are both vascular plants, but the gymnosperms have seeds. The first one is that there is no longer a need for water for fertilization. This is an important advance because we now use the wind. Secondly, there is a loss of antheridia. A loss of antheridia. You remember, these were the protective jackets that surrounded the sperm that kept them from drying out. But we no longer have flagellated sperm in general. There are one or two exceptions. There is a reduction in the size and complexity of the gametophyte. They're getting smaller. They're The gametophytes are no longer free living. And the last of the differences is really one we referred to before, which is that we have 5, the formation of the seed. So when you make your table, moving up from the ferns to the gymnosperms, these are some of the characteristics which you should put in your table to help you understand what has changed in the evolution of this new group, the seed-bearing plants. So I'd like to leave the gymnosperms now and move to what we consider the last evolutionary advance which occurred, and the one which has resulted in most of the plants which you have encountered in your daily lives and most of the plants growing outside here on the campus. Now, this next evolutionary advance, we believe, was originated about 150 to about 190 million years ago, and we think the driving force for this evolutionary advance was the development of extra protective structures for the embryo. That is, we are getting increased tissue to protect the embryo in this next group of plants that we're going to consider. This next group of plants, which is the final group that we're going to talk about as far as evolution, and which will occupy most of the remaining lectures, is known as the angiosperms. And we believe it has evolved as a consequence of the need to develop more protective tissues for the embryo. And we further believe that the angiosperms have evolved from a now extinct gymnosperm ancestor. Now, just like in the gymnosperms, where we said that the word gymnosperm means naked seed, this is gymnosperm. The term angiosperm also is a description of something which happened to the seed. And angiosperm basically means seed in a container. Seed in a container. So I want to take you back to the demonstration we did last week. Here is a female cone of a gymnosperm, and if we turn this cone upside down, the seeds directly fall out. They're naked. Nothing else is surrounding this seed. So these are naked seeds. And this is contrasted to the seeds of an angiosperm, and we're going to talk about this apple in a moment. And if we're lucky, last year we weren't lucky, we are. If we cut the apple open and look in the center of the apple, you notice that the seeds are in the center of the apple, and they are surrounded by this protective structure on the outside. So the seeds are not naked. They are in a container, and the container is the structure which is surrounding the apple. We're going to talk about in a moment where this container comes from. Now, the angiosperms have developed this container, and we consider the development of the container a major characteristic of this particular group. The container, and I'll put this in quotes, which was developed by the angiosperms, you know of as the fruit. Something most of you have eaten or eat every day. The second distinguishing characteristic of the angiosperm is the flower. This group of organisms, which number today about 240,000 different species, is characterized by having a fruit to protect the seed so that it no longer is naked, and having a flower. And I'd like to begin our discussion of the angiosperm by talking first about the flower, because by understanding the characteristics of the flower and the tissues associated with it, you'll be able to understand where the fruit came from, at least where the fruit comes from developmentally, although we may not know in an evolutionary sense exactly how it arose. So let's begin by talking about the flower, and then we'll move on to considering the fruit. The first thing that we can say is that the flower is a determinate, and I'll explain this in a moment, axis along which are arranged a series of organs. Now, what does determinate mean? In our purposes, or in a developmental biology sense, determinate means that it has a fixed period of growth and development. In other words, it's not infinite as far as its growth and development. It has a fixed period. Now, let's look a little more closely at this determinate axis, and in particular, look at the organs which are arranged in a particular series. So the first thing I'm going to do is draw the axis for you. The axis. And I have here now some examples of flowers. And these flowers are essentially a determinate axis. And what I'd like to do is to take apart one of these flowers and to point out to you something which many of you know already. That is the organs which are associated or arranged along this determinate axis. In many flowers, although not all, the most outer whorl or ring of organs, which we'll draw as the lower whorl, are green, and they're given the name sepals. As we move up the axis towards its tip, we encounter the petals. These are the showy parts of the flower. So the next ring or grouping of organs as we're moving up the axis are the petals. These are considered to be sterile organs. And you'll see why we use that in a moment. Now, if we take off all the petals of the flower, there's another one here that's whole if any of you are really interested in seeing a whole flower. Take off all the petals. The next group of organs which we encounter are somewhat brownish or yellowish in their color, although that can change in the many different flowers that exist. So the next group of organs which we encounter are generally not highly colored. These usually are highly colored, although not always. The next group are the stamens. We're going to spend some time talking about the stamens. And if we go up to the very tip of the axis, the very end of this determinate axis, we find the last group of organs which are known as the carpels. Known as the carpels. These are the reproductive organs. The stamens, as most of you know, which we'll talk about more in a moment, make the pollen, which makes the male gamete. And the carpels make the female gamete, or the egg. Now what I'd like to do is to spend some time looking at a carpel, because that's an important demarcator with regard to understanding the reproductive life cycle of the angiosperms. The first thing that we can say about the carpel is that the carpel contains within it prior to fertilization, it contains within it, prior to fertilization, ovules. And remember, we defined an ovule previously as a megasporangium with the megaspore. These are all the stages that you can find inside of a carpel. With the megagametophyte, with the egg. The ovule potentially contains all of these structures. It is the structure with the incipient seed coat. So now let's draw a carpel and put in the ovules and the other tissues which are important in the reproductive cycle of the angiosperms. Carpels generally have three different parts, so we're going to draw a carpel. And most of you are familiar with these parts and have seen them before. The very top of a carpel, we have a sort of a rounded section, although it's varied in its structure. It then has beneath it a quite a bit narrower region that can be much longer or much shorter. And then finally, it connects to an inflated region or an enlarged region down at the lowermost end of the carpel. These regions, as you might guess, have a name. This is the stigma. This is where the pollen lands. This narrow region connecting the stigma with this inflated region is called the style. And this region down at the bottom here, which is enlarged, is called the ovary. That's all that makes up a typical carpel, these three parts. And in all the flowers, these can vary in size and in color and in shape. For this particular carpel, let's put into it 4 ovules. So inside this carpel, we have 4 ovules. And again, these diagrams are in your outline notes, which are available for you to download. Now, let's make a high power of 1 ovule. 1 ovule. Where does this ovule come from? Basically, we have a megaspore. And in this particular case, we had 4 megaspores. Each ovule, each megaspore produces 1 ovule. 1 ovule. Now, what did we say spores grow into? Gametophytes. In this case, a megagametophyte, and because it's the female, the megagametophyte will make the female gamete. And what's the female gamete? Egg. Okay. So let's assume the spore is now N, and it's going to germinate or produce the megagametophyte. In this case, we're having 4 of these going on, but let's look at one of them. If we look at the mature megagametophyte, that is, after all the divisions have gone on and we form the egg, this is what it looks like in the angiosperms. I'm going to draw it for you here. We have 3 cells at one end. At the other end, we have 3 cells with an enlarged central cell. So this is the mature megagametophyte. This is the entire gametophyte of the angiosperms, the female gametophyte. How many cells make it up? So in this particular case, watch my counting here. We have 1, 2, 3, 4, 5, 6, and then this giant cell in the middle, 7. So we have in the female gametophyte the angiosperms, they have 7 cells. And how about the number of nuclei now? How many nuclei make up this female gametophyte? Well, we've got 1, 2, 3, 4, 5, 6, and now this is a really interesting event which happens. In the very big cell, we have 2 nuclei sitting together. So we have a total of 8 nuclei. So this is the complete female gametophyte of the angiosperms. It has been reduced down to 7 cells, 8 nuclei. Think of the female gametophyte in the mosses. Giant structure, millions of cells. Think of the female gametophyte in the ferns. A pretty big free-living structure. This is no longer free-living. Just like you saw in the gymnosperms, the female gametophyte was no longer free-living. Now, the 2 cells that I want to draw your attention to, or the 2 sets of nuclei I want to draw your attention to among these that I've drawn here, are only 2. This cell over here is the egg. That's the female gamete. And these 2 nuclei here are the endosperm precursor. We're going to talk about that in a moment. So this female gametophyte is now ready to be pollinated, now ready to be fertilized by the male. So now let's take a look at the male and see what happens when the pollen arrives and where the pollen is produced. First of all, we said that the stamen is the male gamete producing organ. So let's draw a stamen. Stamens generally consist of 2 parts, and many of you have seen this, and I'll show you some pictures of a stamen in a moment. Kind of a large, often colorful, flat area, which can be hinged, and an area which is attached to a stalk that connects it to the plant. This area here is known as the anther, and this is known as the filament. The anther and the filament. The anther makes pollen. Meiosis occurs here. To make microspores. So if we're making spores inside this structure, what would that structure be? It'd be a sporophyte, but what structure immediately is outside the spores? This is a sporangium. The sporangium. Now, let's say that the pollen is ready to be shed. What does the pollen look like? What does the microspore look like at the time it's going to be shed? And in this case, we'll talk about how it gets over to the female plant very shortly. In general, and I'm going to generalize here again, the pollen typically consists of 2 cells, but not always, there are differences, in which one cell has 1 nucleus and the other cell has 2 nuclei. These represent the sperm nuclei, called sperm nuclei. There are 2 of them now. Very important point. Now, what happens to these sperm nuclei? Let's assume that the pollen gets over to the female, that is, to the carpel. How does pollination occur? Well, as you might imagine, there's lots of pollen flying around outside. All the different species are flying around. Insects are bringing all different kinds of pollen to the plants. The plant has to be able to recognize which is the correct pollen to allow for fertilization. And so pollen grains are covered with proteins, which are part of recognition factors. These proteins are part of why many of you get hay fever, because you're reacting to them in your body and you're sneezing and perhaps other things, your eyes are watering and so forth. Once the pollen has arrived and once recognition has occurred between the pollen grain and the plant, and it's a very complicated series of signals which go on, we get germination of the pollen. Germination of the pollen to produce a pollen tube. And if you could actually see the pollen tube, you would see it. Here's the, let's say this is the pollen grain here, sitting over on here. Here's the grain. The pollen tube grows down the style. And let's make, in this case, only one ovule. This would be an ovule here. One ovule. Pollen grain goes down, and we believe there's an attractant. And the pollen tube goes to one of the ovules. So if there are many ovules, you could have many pollen tubes growing down the stigma and the style. And then we get fertilization. Once the pollen tube reaches the ovule, the two sperm nuclei begin to fuse with tissues inside of the ovule. And I'm just going to draw for you sort of an abbreviated ovule now, and only put on, in the female gametophyte, two of the landmarks. The egg, which is N, and the endosperm precursor, which is 2N. And this is the amazing event which happens, and we have no idea how it happened or what pushed it. Here is the pollen tube. Pollen tube moving down. The two sperm nuclei, and this is the amazing thing. One goes toward the precursor endosperm, and one goes toward the egg. So that we get the following. Here is our ovule again, or what will be the ovule. We wind up with 3 nuclei here. So this is 3N, and the new embryo, which is 2N. This process, where you get 2 different sperm nuclei fusing with 2 separate tissues or zones of the female gametophyte, is known as double fertilization. And it is unique to angiosperms. We have no idea, or some people have some thoughts, but what pushed this kind of double fertilization. We will be spending more time in the next lecture talking about this tissue, which is called the endosperm, and of course more time talking about the embryo. But now what I want to do is answer the question that I posed in the beginning, which is, where does the fruit come from? Where is the tissue that protects the seed? Where does it come from? And the answer is here. Instead of 4 ovules, let's say we now have 4 seeds because fertilization has occurred. The answer is that this tissue here becomes the fruit. So the fruit in most but not all plants is usually only ovary. So the ovary is the new tissue which has developed to protect the seeds, and it is the tissue which leads to the formation of the fruit. Now what I'd like to do is briefly mention to you another aspect. We said that in the angiosperms that we had the development of seeds and we had the development of the fruit, which allowed this group of organisms to dominate the Earth's environments as far as the plants that are outside. A huge number of the plants, a high percent, over 95% of the plants outside are angiosperms. There's one other feature or factor which allowed the angiosperms to become very dominant. And that factor is the fact that the angiosperms have developed mechanisms, which we're going to talk about in a moment, for guaranteeing or increasing the probability that the pollination will occur, that pollen will be transferred. And the way they do this is they develop flowers which have particular characteristics that attract pollinators or organisms. And in return for the organism coming to the flower and getting pollen, the plant gives the organism a reward of some kind, whether it's nectar or other types of things that we'll talk about in a moment. So the angiosperms have been successful as a group because they have developed mechanisms for using pollinators. And when you use a pollinator, rather than having the wind, where we said you require 1 million pollen grains for each successful fertilization, when you use a pollinator, you require only, on average, 6,000 pollen grains to get one successful pollination. This development of a flower and the pollinator together, which has evolved to benefit both organisms, is known as coevolution. And I'd like to now discuss with you a number of coevolution mechanisms which result in a very interesting, successful pollination story. You're welcome to take notes on this, but you don't have to. But I just want to tell you some stories which I think you'll find of interest. I'm going to put the screen down now, but we're not going to show the slides quite yet. The first example of coevolution that I'm going to show you slides of in a moment is a little complicated, and so I wanted to spend a few minutes diagramming it for you before we actually show you the slides. This takes place in figs. Many of you have had figs that you've eaten. Maybe you have it growing in your yard. In the fig, the thing that you eat and you call a fruit is technically something else. So here's an example of something that we call a fruit but is not just the ovary. If you cut a fig open, you make a longitudinal section through the fig, what you find is that the inside of the fig is lined with tiny flowers. Lined with tiny flowers. And if we look at these flowers in particular, they are of one of two shapes or sizes. And I'm going to just draw for you the carpel of these two flowers. In one case, you have a long style. And in another case, you have a short style. These are carpels. A short style. You're not going to be tested on this, but I want you to hear about it. Now, how does pollination occur inside this structure? The life cycle, the reproductive cycle of the fig begins the following way. A small female wasp coming from another fig plant comes to the edge of the fig, and there are some scales on the outside of the fig. The wasp loses its wings and makes its way to the interior of the fig. It's carrying pollen from some other fig that it gotten the pollen from. And as it crawls over these flowers here, it is pollinating them. But what does the wasp get out of this? The flower obviously gets pollinated. The wasp, what it does is it takes its ovipositor, the part of its organ, and it deposits its eggs into the area where the ovary is. And when the eggs germinate, or grow, when they hatch, I'm getting to the plant section here, when the eggs hatch, they develop into larvae, which then consume the fertilized ovary. However, the ovipositor is only long enough so that the wasp can go down through the short style flowers. In the long style flowers, it deposits its egg, but the egg just gets caught in the middle of the style and dies when it hatches because it doesn't have any food. What happens then is that with some time, the eggs hatch, they turn into larvae. The larvae then undergo formation to form wasps, and you have both male and female wasps inside the structure, which we're calling a fig fruit. The males and females mate, and at this point, the flowers inside start to produce pollen. The female gets pollen on her. She has already been fertilized by the male, and she leaves and repeats the cycle again. The males die inside the fig fruit. So for those of us who are vegetarians, when we're eating figs, we're actually eating some vegetable and animal material in the form of the male wasp. So what I'd like to do now is to show you some pictures, first of all, of flowers, and then also to show you some pictures of coevolution. So if we could have the lights down, please. Okay, this first slide is just to show you the parts of the flower again. And again, on the outside of the flower, we have the sepals and the petals. And if we look more closely, we can see the origins of the ovary here. And the ovary is the part that enlarges and forms this protective structure which is around the seeds. Flowers, as you know, come in various sizes and shapes, and a lot of people spend their lives studying it. This flower has often flowered at the botanical garden, and we'll talk about it more a little later on. Here's an example where you can see the sepals and petals are the same color on this plant. And here are the anthers, and the yellow represents pollen or microspores. And if we make a cut down across an anther, we can find the 4 sporangia, the 4 chambers in which microspores or pollen grains are produced. And if we look at them in high power, we can see these chambers with the pollen grains, and you're going to see this in the lab, which are undergoing meiosis. They will be released as microspores and then grow into with pollen tubes. And here is a pollen grain. It's undergoing 1 round of meiosis and then a second division to make 4 microspores, and they are all N. And on this one, you can see a portion of the complicated cell wall outside the pollen grain, which has these recognition factors. When it germinates, we produce this pollen tube, and if you look carefully in the pollen tube, you can actually see the nuclei, the 2 sperm nuclei crawling down. Now, here is the fig that I talked to you about. Here is the fig with the opening here, which consists of scales. And through this opening, the female wasps enter. And you can kind of see, this is a longitudinal section. Here is the male who is blind and the winged female. And the mating goes on. And the process that I discussed with you below or before occurs there. So we have the female going in, the mating, the laying of the eggs, and then eventually leaving with new pollen and also being fertilized. And these are just to indicate to you that there are other factors besides, and they involve levels of CO2, which are important in regulating the flower. Here is a female with her ovipositor, and you can see this is a long or short style. The ovipositor is able to reach the bottom of this particular ovary, but in other flowers it's not. Now, other examples of coevolution that you're familiar with are the orchids. In this particular orchid, if we look at it from this direction end on, what you see is there's a slight hole or opening. The insect enters this because of a smell or perhaps because he or she knows there's a reward, and in order to exit the flower, has to crawl up through the flower's top and in the process gets some nectar, but also gets put onto its back pollen grains. In this case, they're in sacs, and these insect then goes to another orchid and repeats the process where the sacs are removed. The orchids are probably the best example of coevolution. Here is an orchid flower which is meant to look like an insect to attract the pollinator, and in this case it's the female insect. Here's another example of an orchid flower meant to look like the female insect with antennae, with wings, and with an abdomen here. Here's a really great example. Hard to believe that these are flowers because they look so much like the insect which pollinates them. Now, why have the flowers gone through all of this massive evolution and putting a lot of genetics into making these particular flowers that look like the insects? Well, here is the flower. I want to show you how much they mimic the insect. The female of the insect has hairs on her back, and the orchid flower has also produced hairs on its back. And this process, when the male pollinator comes to it, he actually thinks that he's mating with the flower. And we call this process pseudocopulation because the insect mates with the flower. In the process, the flower gets a chance to transfer its pollen, and presumably the male, his reward is that he goes away a little happier. We're not quite sure. This process of pseudocopulation occurs in a number of different species. This is a male grasshopper, and this is a female grasshopper, which is really a flower, and sort of shows you that beauty is in the eye of the beholder. Another example for those of you who might come from the desert or from Southwest where yuccas grow. Yuccas are pollinated by a moth. These are white flowers which come out at night, and the moth actually deposits its pollen in the flower, lays its eggs in the flower, and then the eggs hatch and they consume part of the ovaries, but not all of them. And here's a very interesting example that I want to point out to you for those of you who come from areas that are snowy or like New England. This is a plant which is called skunk cabbage. Skunk cabbage is given this name because it has a volatile, very smelly compound, and this smelly compound is produced because the plant heats up slightly and then in the process attracts insects which are drawn to it thinking that it is carrion, that is rotting meat. And this particular coevolutionary mechanism presumably benefits the plant, although it's unclear how it benefits the insect. And finally, second I want to show you is this flower over here, of which I have an example. This is a flower that's pollinated by marsupials. They crawl over it and they give their pollen chance to be transferred over this flower. And the last slide I want to show you is a question that I want to ask you. We said that the transfer of pollen by insects results in only the need of 6,000 pollen grains compared to 1 million if you use wind. As efficient as this system is, many angiosperms have gone back to wind pollination, and the grasses have reverted to wind pollination. And my question for you is, if pollination by insects, especially through a coevolutionary mechanism, is so efficient and so good, why would angiosperms revert to using wind pollination again, where we have to now use 1 million pollen grains to get one successful pollination? Think about that, and I'll see you again on Friday.